There is a compound that has existed in medical science since 1876. It is 150 years old, and in 2026 it is producing results in cellular aging research that are...
Why have you never heard of it? That is the right question. And the answer involves a patent cliff, a pharmaceutical industry that cannot profit from a compound it cannot exclusively own, and a century and a half of data that's been sitting in plain sight.
Methylene blue. Originally synthesized as textile dye in 1876, used in medicine for over a century to treat conditions from malaria to carbon monoxide poisoning. And now, the frontier of mitochondria.
Before we get into the mechanism, let me tell you what most people believe about cellular aging. Because what you have been told is not wrong exactly. It is just incomplete in a way that changes everything about how you should approach the next 20 years of your biology.
Most people understand aging as where the body degrades, cells stop dividing, the machine slows down. That framework is not incorrect, but it leaves out the most important part of what drives that degradation, and the fact that it may be partially reversible.
The mitochondria, the organelle inside every cell that produces adenosine phosphate, ATP, your body's slow energy currency. Every function your body performs for every repair mechanism, every immune response, every thought you hum.
As we age, mitochondrial function declines, but because the electron transport chain, the process by which mitochondria can convert ATP into habits increasing efficient. They don't increase it efficient.
This is where methylene blue enters. Its molecular structure allows it to function as what researchers call an alternative electron carrier. When the primary electron transport chain is struggling, when those electrons outward, it completes the circuit.
What that means in biological terms, methylene blue may allow aging or stressed mitochondria to produce ATP than they could otherwise. It does not replace the mitochondria. It acts more build the bypass.
In a 2024 clinical review published in the journal Aging, researchers analyzed mitochondrial output in cultured human neurons treated with methylene blue versus neurons, neurons for accelerating and neuron controls.
40% higher ATP production in aging neurons. If that finding holds in larger human trials and the ongoing research suggests the mechanism is real, the implications for neurodegenerative disease, cognitive... The second mechanism is oxidative stress reduction. When lactose from a methylene transport chain, they react with oxygen to form reactive oxygen species, free radicals. These are the molecules that damage the membranes and DNA.
Two mechanisms: increased ATP production, reduced oxidative stress, both emerging from the same molecular action, and both directly relevant to the primary drivers of cellular aging that researchers have spent 30 years trying to target.
The brain is the most metabolically demanding organ in the body. Neurons acquire packs when one of these painting petrins truncadians, when mitotanes in neurons, specifically the cognitive physicians, and perform repair. In early studies on methylene blue and cognitive function, specifically models of Alzheimer's disease, we stuck on reductions in yeast enteroprotein. The mechanism for autophagy contained is superoxynuclear but accelerated aggregation. Let me tell you about a patient I will call Margaret, 62 years old, no neurodegenerative diagnosis, but experiencing what she said was fog. The kind where the word you want is there, but the retrieval takes a half-space and honk whenever used to. The kind where it ends.
Margaret was not clinically impaired. Standard biomarkers were within range, but her subjective experience of cognitive performance had shifted over 3 years. She came in asking what she could do, not what diagnosis she had, but what she could actively change.
We discussed the emerging research. We reviewed her baseline, and we talked through a low-dose oral methylene blue protocol starting at 0.5 milligrams per kilogram of body weight 3 times per week, not a prescription, a monitored supplementation approach and transesperate.
At week 8, Margaret described what she called a return, not a transformation, and retrieval lag at short. Morning clarity extended past noon. Sleep quality, measured on a wearable, showed a 70% improvement in deep sleep duration. Correlation, not causation, but notable.
I want to be precise here. Margaret's experience is one case. The clinical literature on methylene blue and human cognitive function is promising but not yet conclusive at the population level. Larger randomized controlled trials are underway, but we have our mechanisms for addition or biological clearance right now. The safety profile of methylene blue low doses is one of its most significant advantages. It has been used clinically in humans since the 1890s. It is on the World Health Organization situs of anemia. But therapeutic doses of tronithin. There are contraindications. If you are on certain medications, SRIs, higher dose carries a risk of serotonin syndrome. This is reverse direction, and it requires a conversation with your physician for any supplementation. Additionally, individuals with G6PD deficiency, a genetic enzyme disorder, should not use methylene blue. Your physician can screen for this with a standard blood test. These are not reasons to avoid the research. They are reasons to approach it through a medical conversation from a supplement. Beyond the neurological research, the cellular aging mechanism of blue is also being explored in dermatological applications. Skin cells, high neurons, are Heineken study published in Margaret human skin cells significantly in cell lifespan.
Cellular senescence, sometimes called the zombie cell problem, cells that often dividing but have not died accumulate with age. They secrete inflammatory compounds that exudate the surrounding tissue. Methylene blue in that 2 to 16 study
What the compound appears to do across neurons, skin cells, and the broader cellular models is address the upstream problem, not the symptoms of aging. The mitochondrial inefficiency, then, is that the leavenly in a supplement symptom. For context, compare the mechanism of the most heavily marketed mitochondrial supplements currently on the market. NA plus precursors, NMN, NR. These work by replenishing mitochondrial function, but does not directly address the transpostor. One additional point that the research community discussed: pharmaceutical were approximately $12 to $18 per gram. A standard low-dose protocol uses milliliters per session. The monthly pot to follow $12 and $15. What does the current evidence support in practical terms? Low-dose oral methylene blue, 0.1 milligram per kilogram, 3 to 5 micrograms with potential methylene support strategy for adults or can for vaccine post-immunization. The question Margaret asked, and the question I imagine you are asking, is What does this feel like if it is working? Because the problem with mitochondrial health is that it is invisible until it is not. You do not notice your ATP production being declining. You notice that the morning...
The most consistent reports emerges in the ongoing low dose of mitochondria are improved cognitive clarity and the force exchange of statute and ultra sleep metrics and something deep in REM sleep during.
The sleep connection is particularly interesting. Deep sleep is the phase during which the glymphatic system clears metabolic waste from the brain, including the beta amyloid plaques associated with Alzheimer's. If improved mitochondrial function, the downstreams as well be necessary. Active trials as of 2026, FA standardized mentality in the blue and cognitive aging in adults over 660 is excellent. A separate neurology trial examining mechanism weakness and photoelasticity.
Step 3. If cleared, pharmaceutical-grade methylene blue only, not industrial-grade, not aquarium-grade, and purity determine safety. Step 4. Start at the lower end of the dosing range, 0.5 milligrams per kilogram, 3 times per week.
I want to be clear about where methylene blue fits in the hierarchy of health interventions, not the foundation. Sleep quality, zone 2 cardiac exercise, protein adequation. Methylene blue is an amplifin to Miadol foundry. I'm 14 months in, and I'm holding to protocol. Subjective cognitive markers have held. Her physician is separate cohort 2 extended conflated and victorizes and wearable data. A compound that has been in medical use for 150 years. That costs less than $50 or mechanism that researchers now consider central to the aging process. It is noversin. It does not withstand to amplified. As a clinician, I am cautious by nature. I do not recommend things ahead of the evidence. What I will say about methylene blue is this. The mechanism is real, and safety history is long now. Current cheap is encouraging.
What to do this week? Talk to your physician. Specifically ask about mitochondria testing, whether methylene is for your baseline. Ask about contraindications. If you are any extremist things with the target medications, that for indications that conversation sense mind that should.
This video changed how you think about mitochondrial aging. Share it with someone over 45 who is paying attention to their long-term health. This is the kind of information that takes years to move through clinical channels. Word of mouth is faster. Subscribe to Vital Years. Every week, one mechanism, one study, one protocol that your standard annual physical will not cover. Hit the notification bell. The next video drops Tuesday.